published two most important that I'm really proud of these authors. The first one is we developed a tool that allows you to do multi-round of multiplex immunolabeling in whole mouse brain. And this is really important because sometimes you have a disease model mice, or sometimes you have a really precious sample, and you really wanted to understand every part of it. Then you have to do like, you have like 10 different biomarkers you want to identify, but you cannot label them at once. Now with this protocol, HIP clear, you are able to do that as many times as you want. And the second major paper we accomplished this year is the collagen expansion microscopy method applied in collagen abundant tissue, meaning that in addition to brain tissue, you are able to do expansion microscopy in a lot of different tissue types in the body. So in our lab, we have technology developed. My group, they are working on different aspects like beauty microscopy, making the tissue transparent, and develop new labeling technique, and also the deep learning analysis. And we have collaborated with a lot of amazing scientists, including neuroscientists and the scientists work, the material scientists. So a lot of the work actually, like the Professor Lo just mentioned, is published by this drug delivery group, and they're superstars and they're really amazing. Their paper, they have this paper publishing strategy that I'm really admire, that they have this factory type of publishing. I couldn't really understand like what's the difference between different papers actually. So they change different molecules, they change different design of their materials, and go through the same series of experimental tests and just do the publishing. So it's kind of factory, but it's not as fun as what you do now. So don't envy them. Okay. And recently our lab also working on digital pathology works. So we just got a first patent approved that we're applying deep learning on analyzing HER2 dish pathology samples. So this is how my lab, my lab looks like. So we have this tissue engineering team, and microscopy team, and deep learning team. And the reason why we need so many people is that in order to give you something this and useful, we need quantitative data out of it. We really need, to do the entire pipeline in our lab. There's no one else to help you in, some points here or some point there. You have to do everything by yourself. And the reason that we are so dedicated to do this is imaging is one of the key measuring method in how you evaluating the disease progression, or what's the efficiency of your drug treated your animals. And so this is one. Of course, there's other evaluation method to help you evaluate if the animal is getting better or learn better. But without imaging, you won't be able to really quantify the difference between the control animals. And there are so many ways to do image. There are like MRI way, and also like electron microscopy. They are all fantastic tools. But the problem with MRI and the EM is that you don't get to label specific protein directly. You might be able to distinguish different part of the body, or like different neurons based on their shape. But if you look at this picture, for example, is there anyone know that what's the black spot here? No, right? I mean, for the fly expert, probably. But so this is mitochondria. Actually, mitochondria is everywhere in the brain. This is the really small chunk of the fly brain image, and The other reason that we wanted to find another way to image this sample instead of MRI and EM is like, doing MRI is really easy to get a whole picture, but the resolution is relatively low. Okay, it's 200 micron each pixel for if you image with the best MRI machine in the world right now. But 200 micron is still very big. The fibrin is about 500 micron, and there are like 100,000 neurons there. So obviously you can't really see each single neuron from MRI, right? But if you want to do everything with EM, it's also impossible. I don't know how many of you know that to image a fibrin through serial EM imaging, how long does it take? More than a year. It's 16 months to image just a single fibrin. So it's taking too long if you wanted to see something comprehensively and detailed enough. So, so this is what people need. We need to visualize whole organ in order to understand our body or what happening in the animal systematically. And we want to be able to identify the target protein or where the drug goes. We want to do it fast. We don't want to take like 2 years to just finish one set of image. And we also want to correlate this data with the treatment behavior. That means we need a really fast way to analyze them. So this is why my lab focusing on developing microscopy tools. Optical microscopy now able to allow us to do whole brain multiplex, as I just showed you. And we can also do super resolution image. And we can also do live imaging. That's something these guys can do. So we can label neurons and monitor their activity at the same time when they're behaving. That's what Professor Low is doing right now. And a lot of neuroscientists do every day in the world, in every lab. Okay, so how does the brain looks like if you look at not in pieces but in an intact brain? So this is a video that's been done for many years now, and you can see there are two parts in the brain being labeled. The blue one is the dopamine neuron, and the red one is the acetylcholine neuron. So the reason we did this image is there are collaboration between Professor Wu in Chang'an University. They are doing addiction study, and they found the dopamine neuron have some, will do something weird to the acetylcholine neurons after the mouse get addiction. Okay, and on the right side of this, I hope this image convinces you that this is the super resolution optical microscopy acquired in the fibrin, and it's showing the same detail as the electron microscopy gave you. Like you can see all the arches on these synaptic terminals. So how do we do this? We have to build different tools, right? So the first part is the large organ one, and before you have these two, before we have this tissue clearing, the way we do imaging on mouse brain is like this. So we cut the brain into small pieces and quantify the cell in this area. And this might make sense to a lot of oncologists, like if they are not doing brain, they are doing tumor tissues. But it doesn't really make sense to me if we're as a neuroscientist, because you don't want to only see the soma, you want to see the entire neuron, right? Especially I was trained as a neuroscientist doing flies. In fly area, nobody doing sections at all. Like before I started working on mice, the idea of tissue sectioning basically is not in my head at all. So when I first see this image, I was like, no, I wanted to see where this cell goes, right? And how the 3D structure of these thalamus areas. So, striatum areas. So this is the projection image of the previous video you see. So by doing the whole brain, you are able to see the soma of each dopamine neuron and see their axon terminals and see where they're sending to different area of the brain. And this could be done because of the tissue clearing, which is another tremendous work that Professor Zhang did like 30 years ago. Okay, but now people are able to do this process in a daily basis. So the idea to make brain transparent is that if you don't do it, the light from the optical microscope cannot get into the sample, and so that you cannot get the signal out of this beautiful organ. And you can totally do this on different kind of organs. So this could be done in intestine, kidney, liver, heart, lung, and spleen. If you wanted to see them directly in live, like you can come to my lab or visiting Brain Research Center. We have the real sample there, showing you that how transparent this organ can be. And we also developed some pattern from doing this experiment. And this job can be done in many ways. If you go through Google Scholar and just type like brain clearing, there are like so many protocols. But there's one lacking, key step is that nobody can ever do this process from a FFP embedded sample. And why this is important? Because every hospital in the world is using wax embedding to preserve their precious biopsies. This is the only way that you can put the sample for like 100 years, and you don't need a fridge. If there's earthquake, if there's power outage, I believe some of you might experience already, you don't have to worry about it. You don't need a fridge. And it's very solid that you can't really break it even there's an earthquake. If everything just falls down to the floor, it's fine. Not like the monitor in my lab a few months back. Okay. So what we did is we found another way called HipClear, which enable us to retrieve the sample from the FFP embedded brain and allow us to do the multi-round labeling as well. And here is just showing you that we can image the brain and register it to the standard brain so that we can quantify the expression of different kind of cell that in each brain area. And this is the process of it. It's not super, complicated, but still, the first part is the FFP blocks, and then we have to do like dewaxing, rehydration, and delipidation. And the main development of our paper is on this part. So retrieve the epitope from this wax embedded sample is really tricky. But after this, after we retrieve all the epitopes, we're able to do like immunolabeling and then use light stream microscopy to do the image. So here just one example to show you that we're able to do 6 rounds of immunolabeling and to label 6 different biomarkers. And the reason why we wanted to do these 6 rounds is you need to label them with specific marker like CFOS activity marker so that you are able to know how many dopamine neuron is activated, how many acetylcholine, how many like GABAergic neuron is activated during your experiments. Okay, and one exciting thing that we did when we are doing revision is that this process can be applied on the sample that been fixed in formalin over 3 months. So one key reason to do this is in the brain bank, a lot of sample will be put in the formalin over several weeks before they do the further step, like FFP embedding. So it's really important to show people that we are able to retrieve protein signals out of this type of sample. Okay, so this is organ imaging. What about the super resolution? So I believe many of you already know that the diffraction limit of microscopy is around 250 nanometers. And in order to distinguish things smaller than this, let's say like 2 points, and they only have 100 nanometer from each other. In the past, we can only use electron microscopy, but now optical microscopy can still do it. If you look at the paper 1 year ago, there's a paper called One Microscopy, you can Google it. They use super resolution combined, they use expansion microscopy in addition with SRF together to get 1 nanometer resolution. And they're able to do protein structure. So I know many of you are like doing protein structure in the lab, and this is something new you can probably try. So this is how neuron will like if we do super resolution on them, and we're able to see different vesicles around each synapse. So this is really fantastic, and we can apply it to many different tissue types. Like this is a chunk of human brain. You see these are astrocytes and link the neuron to the blood vessel together. And this is not a very big piece, but still like a millimeter or something. So it's big enough, allow us to still see pathological changes. And brain is easy, actually. There are so many people working with brain. But skin and a lot of collagen abundant tissue is hard, especially a lot of oncology, like tumor tissue. They have a lot of ECM, they have a lot of collagen in there, so it's really hard to do super resolution or like expansion microscopy. So we collaborate with Professor Lin Songran, whose name is on the hallway actually. If you go, and he's the first winner of the So he's expert in dermatology, and we collaborate with him and develop this new tool called co-EXM, collagen expansion EXM, allowing you to see all the detailed neuromorphology in skin or all kinds of like collagen abundant tissues. And if you're interested, you can Google this name and the journal name, and you'll see a lot of more details. And in order to do this, we have to have image tool, right? So this is more like class-based. If you wanted to do image quick, you need to use camera-based tools like multi-point confocal, which is spinning disk microscopy, or the light stream microscopy, because these guys doesn't do scanning. These guys, the confocal you use in the daily base, do scanning, right? I can give you a rough number. These guys have image speed 1,000 times faster than the confocal. So imagine that if you have this tissue slice that have to take you 2 hours to image under confocal microscopy. Now only need several seconds under light sheet or spinning disk. So this amazing speed allows to do a lot of things. So we basically built light stream microscopy in the lab. So if you're interested if the students are interested in doing more engineer work, they will join this group. Okay, we can, this is just like Lego, everyone can do it. They just have to learn not to break it. I'll just show you how they look like. So this is the animation of one of our microscopy. So there are like many small pieces, right? It's just like the Lego that my son just did, tore apart yesterday. Okay, so so I'll just quickly go through this because we don't have so much. I want to focus on the last part. So the reason why we need deep learning on doing the analysis here, there are several reasons. First thing is the image size is so big. There's never enough hard drive in my lab. Every year we have to, specifically find some budget to buy new storage for this new image. So for each frame, it will take 500 gigabytes. And more terrifying thing is that this data can be generated within an hour. So I'll give you a rough number. I don't know how many of you know Allen Brain Institute. Maybe not too many. Okay. So Allen Brain Institute is private funded, and they're doing the most crazy